Hello everybody, my name is Brandon Toyer. I am a senior geology student here at Augustana, and my senior project is entitled Variety and Identification of Theropod Teeth from the Early Jurassic Hansen Formation of the Transantarctic Mountains in Antarctica. This project was made plausible by Augustana College, the National Science Foundation, and the Field Museum in Chicago, Illinois. So just to give you a basis of how this project started to get going, I had an internship 2 summers ago at the Field Museum. It was actually a 2-summer internship awarded by Dr. Hammer. And the first summer I spent at the Field Museum beginning this project, and then the following summer I spent it at Augustana College continuing my research as well as helping downstairs in the preparation lab. That's the Field Museum right there. That's the view where I eat my lunch every day. It's a great view. This is me at my fossil preparation station, cheesing. And right here is me and a coworker at the time, Sarah Gibson. She's out at University of Kentucky getting her doctorate in paleontology. This is also a fun thing. This is Sue, the big dinosaur at the Field Museum. That is a cast of her foot. And right there is a juvenile T-Rex foot. So you can just see the difference in growth right there. So some background on what I'm going to be doing with my project is focused on. The first thing is the specimens that I was given by my supervisor, Peter Makovicki, at the Field Museum were encased in a siltstone matrix from the Jurassic Hansen Formation. And this is from the same bone bed as Cryolophosaurus elioti was found in. This siltstone was incredibly hard matrix. It's very difficult to remove the specimens from it. Next, isolated theropod teeth cannot be identified unless compared with in situ teeth of the associated jaw. So what do I mean by that? That means I can't have an individual dinosaur tooth and say, based on some measurements or images I take from it, I can't say it's definitely this. I have to compare it with teeth that are already within the jaw fragment or with other teeth that have been identified as definitely it is part of that taxon. Thirdly, the teeth analyzed in the study were found by ours truly, Dr. William Hammer, in 1991 within the Transantarctic Mountains. So here's an image of Antarctica. The Transantarctic Mountains run southeast to west right here. And have further visualization of it. This is an illustration created by Nate Smith, an alumni of Augustana College and who is now a professor of paleontology. And this is the Transantarctic Mountains right here. This is a blown up image of it, and my specimens were found in this general region. It is important to note that Cryolophosaurus elioti was found in the early Jurassic part of the Mesozoic. This is a pivotal time for theropod diversification. So just keep that in the back of your head as we progress through this research project. So, in this study, where this is going to be about the how, the what, and the why of the whole project, I had 6 unknown teeth from the Jurassic Hansen Formation, and my goal is to organize them into distinct morphotypes based on 3 parameters. The first one being anatomical features, the second one quantitative analysis, and the third being qualitative analysis. Quantitative is looking at it numerically, we'll look at some graphs, and qualitative is we're going to be looking at the structures of these teeth. Now, research on theropod teeth, why am I doing this? It's pretty underdeveloped because teeth are not as informative as other skeletal features of dinosaurs, such as like the head, vertebrae, femur, but there are a plethora of teeth out there. So if we can really push teeth identification, we can really unearth a lot of valuable information on theropods and their lifestyles. And more importantly, we can understand some biogeography of that time, which is a huge important factor at looking at paleontology. Also, this is a map of the Earth during the Jurassic time period. So, it looks a little different than it does today. So, the first thing I had to do, Dr. Makovicki gave me a bunch of blocks, rocks like this, probably about the size of your fist, and sat me down. I learned how to prep. It's an awesome time, very relaxing. And you put the block right here. You look at it through a microscope, so you can really see the topography, the details of all the teeth, because these things are only a few millimeters to a few centimeters big. And then you use this microjet right here, and it's essentially the size of a pencil, and it's a jackhammer, very fine detail. And then this thing blows all the excess matrix that you chisel off away. these are 5 of the teeth that I had. I took this image, and after I took this image, Dr. McAfee gave me an additional block and was like, here you go, here's another one. So that took me another 8 days to do. So I apologize for not having a picture of all 6, but in fact, I do have the casts I made of all these teeth, and I'll pass them around so you guys can see them. The casts are these, and then there's 2 additional ones of E and F. You'll see as they get passed around. But, so I removed these teeth from the matrix, from these blocks of rock, and then I had to take some anatomical measurements from it. There are a lot of parameters on this, but don't get overwhelmed. There's only a few that I really focused on. AL, the A4M length, it's pretty much tip to tip of the tooth. The crown basal width, which the crown is the part of your tooth that connects to the gum. So we're going to look at the width and the length of that tooth, of each specimen. And then as well as the denticle density on both the lingual side, which is towards the inside of the mouth, as well as the labial side towards the outside of the mouth. Now denticles, you can really see it and feel it on the specimens that are being passed around. Imagine like a butter knife. Theropod teeth, they have serrations like a butter knife on both the front and the back. It helps with slicing. Because if they didn't have it, the teeth would get stuck and they'd crack off. So it's a pretty cool anatomical feature. So when I had all these anatomical measurements of my 6 specimens, I had to conduct some quantitative analysis of them. I mentioned this earlier, morphometrics. Morphometrics is the quantitative analysis of form. It's a concept of shape and size. So we're going to be looking at these measurements that I've taken and formulating them into some morphometrics. Now how do we do that? We use this program called PAST. It's a paleontological statistics program. And it allows me to illustrate all the dimensions and how each of these samples relate to each other. How do they relate to each other, you ask? By clustering. Clustering, it's essentially on the graph that you'll see in the next slide. It's how all of these teeth, all the parameters that we measure, it's how they're related to each other, so they will locate on the same portions of the graph. You'll see in a second. So the morphometric data, this is the graph that I made. And all of these varying colors are different dinosaurs, all throughout the Mesozoic. I pretty much went through a bunch of journal articles and took away dentition information as much as I could that were following the same measuring guidelines as I previously explained in that, this about 2 slides ago. Now, like I said, these are the clusters. You notice how each of them create these polygons right here, enclosing all of the other points. These are the clusters that I was referring to. Each of these points are also an individual tooth. So each point on this whole thing are a bunch of teeth. There's a ton of teeth in the whole thing. I probably had like 150 to 200 that I put in. Because in this type, and when you're looking at morphometrics, the higher the sample size you have, the better it is. So the more, the better. Now, unfortunately, PAST is somewhat limited, and it doesn't really create a legend for when you make these big graphs, which is it's a free program. You have to give and take. But I just put some pictures here to show you where each of these clusters, like what types of dinosaurs there were. Right here, towards the first and second quadrant of the graph, Tyrannosaurus rex is the brownish one right here. But all on top of that, overlaying with that, are all other massive carnivores, other theropods, bipedal. Big things are at the top of the food chain. And then right here, we have a lot of scavengers, a lot of smaller theropods. Velociraptors, I have a Dromaeosaur right here. They clustered towards the origin and a little bit below it. And then out here is an outlier, is a Troodon. Troodon have they have their own unique teeth. They're kind of an outlier from the rest of looking at all that dinosaurs. They have unique formations on their whole thing. So that's why it outlined away from everything else. And my samples were right here. It's difficult to see, but in there's a little gray polygon. Those were sample sizes A, B, C, and D. Now I said I had 6 sample sizes. A is the smallest, and it progressively gets bigger all the way to F. So E and F are the 2 largest of the samples, while A, B, C, D are smaller. They clustered right here, right on the origin, on top of Dromaeosaurs, and on top of all these smaller scavenger theropods. E and F located very closely to the top dog, top food chain theropods that we probably all think of when we think of big scary dinosaurs. So after looking at that, I was posed with the question, like, why are they clustering in two different locations? I kind of went in this with the assumption that they're probably all going to belong to the same species, but in fact, they weren't. It's either between two things. It's either they're different species, or B, they belong at different phylogenetic stages. Phylogenetics means different ages, so like a baby or an adult. So we had to look at the qualitative analysis, meaning let's look at the structures. Specifically, we're going to focus on the enamel. We're going to look at the enamel structures because in theropods, enamel structures can form in 6 different ways. So we can look at these enamel structures and compare and contrast how the structures of all 6 teeth relate. If they're the same, then we can kind of indicate that they're probably the same species. If they differ, then they're probably different. The enamel patterns are referred to as, in the paleontology world, as Schmelzmusters, a term describing the combination of enamel type and enamel features. I mentioned this earlier, and it's important to note again that the Schmelzmusters cannot be used to identify specific dinosaur clades. They can only be used to compare and contrast and in turn accept or reject a common species. And the qualitative analysis is a destructive process. So molds and casts of the specimens were made by myself, and that's what's being passed around in the class. How did I take my qualitative analysis? By using a scanning electron microscope. A scanning electron microscope, we were looking at the enamel at about a range from 12 to 1,500,000 times magnification. And a scanning electron does that by, you put the right here is a square chamber, it pulls out, you put your sample in there, and then you turn the microscope on, and it bombards it with electrons. And based on how those electrons reflect off of the surface is how it creates that topography that shows up on the computer. And I have some samples of those, and it's pretty neat. There's some gold alloy plating on them, and that allows an easier time for the electrons to reflect off and get a more vivid, detailed image. So, how did some of these images turn out? These were some of the best images I had for each sample. The left column right here is a specimen. The middle column is the longitudinal axis, and the right column is the transverse axis. Now, if you're imagining this pen is like a tooth, it's sitting in your mouth like this, the longitudinal axis is you're going to be looking down from this way. So I envision it as more of the y-axis, and as the transverse axis is looking this way, so kind of like an x-axis. So you have some sort of a frame to put this in. Now, what's interesting, these images are a little washed out, but I apologize. We were magnifying it about 1,500 times for these because these are the smallest of samples. But on the longitudinal axes, A, B, C, and D, that cluster that formed right on top of those smaller theropods, is showing a parallel crystallite enamel structure, indicating that the enamel grows in bands like this, all down this axis. While on the transverse side, it has a columnar enamel structure, meaning that at the bottom right here, which is referred to as the enamel dentin junction, the point where the enamel and the tooth meet, it kind of forms arrows towards the bottom and runs more vertically, while on the longitudinal side, it runs more horizontally. On this next image, you can really see some nice images of it. Right here on the D longitudinal, you can really see some beautiful lines going right across. You can definitely see the parallel habits of it. And then for the transverse, towards the bottom, and actually at the top too, you can see these arrows formulating. But like I said, C and D and A and B all show the same structures on both axes. Now the difference was between E and F. You can really see some strong differences, especially in the longitudinal side, where we're going to be getting some basal unit enamel structures. It's kind of like a bunch of polygons smashed together to form this sort of structure. But on the flip side, on the transverse, it shows a parallel crystallite structure as it did on the longitudinal axis of specimen D. So at this point, I'm relatively confident that these are two different species, depending or based on the separate clusters on the morphometrics, as well as the different enamel structures on the axes. But just to really solidify that idea, I took all the parallel crystallite enamel structure images for each one, and I inputted them into a program called ImageJ. ImageJ is a software program that allows you to measure. You input a photograph, you enter in the dimensions of the photograph. Like I would put in, for example, 1300 times magnification and 30 micrometer width of the image. And then there's some tools in there. I just used the basic one of just a measurement tool, a distance, and I measured the distance between each parallel crystal incremental band. Meaning, you know how I was explaining that each of the parallel crystal or parallel enamel structures grow like that? I was measuring the distance between them on each of these axes that had this type of formation. And the result is I got an average distance where E and F were about 5.1 micrometers thick, as well as the A through D were about 4.2 to 4.6. So there's about 20% variation between those, really solidifying the conclusion that the pair of two larger teeth is speculated to belong to Cryolophosaurus elioti, and the other four belong to scavenger. Like I said, it's speculated because I can't wholeheartedly identify that these teeth belong to this dinosaur, but considering that these teeth were found in the same bone bed as Cryolophosaurus, as well as in the clusters formed over the area where all the other large carnivores were large carnivorous theropods, I can speculate it. It's a strong feeling towards it. And the other four are speculated to belong to some sort of scavenger, one because where they plotted on the clusters, as well as there was some research done a few years ago, and as well as some conclusions derived when they found Cryo, that he was found dead. He was at the point of deposition, he was dead, so he was being buried and he was been long dead. So that would give ample amount of time for some sort of scavenger to come to feed. And so with the successful implementation of quantitative and qualitative analysis, theropod teeth can hold more significance and morphological understanding. With this type of methodology, we can take the plethora of teeth that are available by institutions all over across the United States and really start assigning them to specific taxon, and that will in turn really open up the gates for we can look at feeding habits, biogeography, like I said, and other basic features of the fauna. My acknowledgements, I first off have to thank Dr. Hammer of Augustana for giving me this internship, for helping me along the way, for allowing me to TA his class, being a great friend, everything. Thanks, Dr. Hammer. Dr. Pete Makavicki, Akiko Shinya, and Betty Harmon, they were all employees, and they worked in the upstairs of the Field Museum, all helping with different parts of my project, and in the rest of Augustana's faculty in the geology department. Dr. Wolf, Dr. Strasser, Susan, Gail, Diana, and Josh for when he was here the previous 3 years. Thank you so much. And as well as the National Science Foundation for funding my project and giving me the money to conduct all my research. Here are my references. Does anybody have any questions? It's not Augustana.
I know, but it's trying. That's from 1977 when I was a graduate student.
Any questions?
One point, I agree with all his conclusions except dromaeosaurs and velociraptor are actually the same thing, and they're not scavengers.
I was just speculating that they were
But,
scavengers, they're smaller.
no, they got the big claw, come on. But they're smaller theropods that were scavengers, had teeth like them, so that's what
My journal articles are probably
yeah. I mean, because they were late Cretaceous.
Yes, Dr. Strasser?
On your morphometric data, what were your variables that you plotted?
Okay, so this is going to take a second to describe. So how this morphometric data works is it first converts a linear relationship of each of the variables that I inputted in. A few slides before this, I talked about like the AL, the denticle density, the crown basal width, and all of that. So it takes all of those parameters and creates a linear relationship amongst each individual specimen, and then in turn takes all of those canonical variables and turns them into principal component analyses. So taking all of those individual data points, turning them into a linear relationship, and then looking at how those linear relationships relate to each other. And then that's how you get it. So components 1 and component 2 are sort of a conglomerate of how all the variables relate to each other. Does that make sense?
I think it does. The reason I asked is because I was wondering if you could look at other variables, or now, if you're lumping these together, tease them out, it might teach you something.
The component 1 and component 2 are like 99.9% of the time the best, because depending on how many variables you input at the original state is how many components you have, and the lower you go on down the component, like 1, 2, 3, 4, 5, the lower you go down, the less statistical significance it has, and you're not going to get as much diversity. Like they'll all, eventually, if you look at components 4 and then components 5, it'll just make one linear relationship down, but it doesn't hold much statistical value. As far as to why it does that, I'm not sure, but that's how it does it. Yeah?
Yeah, I understand that, like, with the teeth, as you're saying, it's not like forensics where you can't just like straight up identify the teeth record with people and stuff like that, because we know that this is over millions of years old, obviously. But with this sort of approximations that you made, would this information be useful if, in the case of where you're sort of identifying certain dinosaurs, or maybe just maybe certain types that might have existed at certain periods? I mean, it may not be able to narrow down what the exact type of mammals are, but maybe narrow down the environments or sort of areas of, where past dinosaurs lived.
So you're asking,
Yeah.
can I use like this methodology to derive like how the environment that this animal lived in?
Yeah, or at least give you an idea. Or would that not be helpful?
For carnivores, not as much as herbivores, because carnivores just eat obviously meat. So I guess like we could get some sort of rationalization on like maybe the size of the things they were eating, and based off that kind of get something. But like definitely with herbivores, you can kind of see what kind of plants they were eating based on their dentition, based on if they had gastroliths on the inside of them, those rocks that kind of jumble everything up. So with this, I would say not as much.
All right.
Dinosaur teeth are much less useful than mammal teeth and fossil.
Okay.
Mammal teeth give us a plethora of information, because the morphology is just so outrageously intricate. Dinosaur teeth, they're kind of dull, so to speak.
Any other questions?